T cells are the lymphocytes responsible for the cellular response. The T in T cell stands for thymus, a gland located above the heart where the T cells develop. Molecules that are a part of the body are considered by the immune system to be self molecules. All other molecules are considered to be non-self. During development, T cells in the thymus are tested to see if they react to self molecules or non-self molecules. T cells with antigen receptors for self molecules are destroyed. Only the T cells with antigen receptors for foreign molecules are allowed to develop. The developing T cells interact with thymus cells that contain both MHC1 and MHC2 molecules. Some of the developing T cells are reactive with the MHC1 molecules. These cells develop into cytotoxic T cells. Other developing T cells are reactive with the MHC2 molecules and become helper T cells. Let's look at the roles of cytotoxic and helper T cells to see why each reacts to a specific MHC molecule. Cytotoxic T cells kill cancer cells and cells infected by viruses and other pathogens. Recall that all cells express MHC1 molecules on their surface. As a newly synthesized MHC1 molecule moves to the cell surface, it picks up a fragment of one of the proteins synthesized in the cell. If the cell is infected by a virus, the fragment could be from a viral protein. The viral protein fragment is carried to the cell's surface, where it is presented to the cells of the immune system. The interaction between an infected cell and a cytotoxic T cell is enhanced by another cell surface molecule called CD8. CD8 interacts with a part of the MHC1 molecule, keeping the two cells together while activation takes place. When a cytotoxic T cell is activated, it releases perforin, a protein that forms holes in the membrane of the infected cell. As a result, water and ions flow into the cell, eventually causing it to rupture. Helper T cells assist in the activation of cytotoxic T cells and also help recruit B cells to fight an infection. Remember that only antigen-presenting cells express MHC2 molecules on their surface. Helper T cells interact with MHC2 molecules on macrophages and B cells. Macrophages engulf and digest pathogens like bacteria. As a newly synthesized MHC2 molecule moves to the cell's surface, it picks up a fragment of the digested bacterial proteins. The bacterial protein fragment is carried to the surface, where it's presented to the cells of the immune system. The interaction between a helper T cell and an antigen-presenting cell is enhanced by CD4 molecules, which help keep the two cells together while activation takes place. Activated helper T cells secrete several proteins or peptides, collectively called cytokines, which stimulate other lymphocytes. Some helper T cells secrete the cytokine interleukin-2, which induces the clonal selection of both cytotoxic T cells and B cells. Cytokines also stimulate some helper T cells. For example, antigen-presenting macrophages secrete the cytokine interleukin-1, which activates helper T cells to produce interleukin-2. This is an example of how the different cells of the immune system work together. Let's see how well you understand how cytotoxic and helper T cells work. Then we'll look at the role of B cells and antibodies. Drag each label to the appropriate location to identify the different cell surface molecules. Click Submit to see if you're correct. Sorry, that's not right. Yes, that's correct. Cytotoxic T cells have CD8 molecules on their surface, whereas helper T cells have CD4 molecules. All cells express MHC1 molecules. Only antigen-presenting cells, such as macrophages, express MHC2 molecules. Now, drag each T cell so it can be activated by the appropriate target cell. Make sure the surface molecules are lined up correctly. Click Submit to see if you are correct. Sorry, that's not quite right. Yes, that's right. Cytotoxic T cells interact with antigens presented by MHC1 molecules. Helper T cells interact with antigens presented by MHC2 molecules.